video we will continue with building our understanding, our practical understanding of FBA. We will look at how you go about troubleshooting an FBA. So, the classic problem is you have a model, it does not show growth, how can I fix it, right. So, what are the things that one might need to watch out for, what are the possible sources of this error, and we will also look at the concept of ATP maintenance flux and this very interesting concept known as flux variability analysis, which tries to tell you what are the minimum and maximum fluxes that any reaction can admit or any values a flux can take under a given set of constraints. So, you now understand the concept of a gap, a blocked reaction and dead end metabolites, and also how to put a band-aid fix for dead end metabolites. Right. So, the most important thing you need to really worry about when you are performing FBA is the first thing you will encounter is you build a model or you download a model and you do optimize CB model, you will find that you get 0 growth rate. How do you fix it, right? So, let us say we say common problem that everybody encounters. Practically, what do you think this could be because of? What are the reasons that might give you 0 growth rate? So, the first thing would be are my median constraints making sense or not? So, one way of looking at this, the obvious reason why this happens, immediate reason consequence of this, or this is an immediate consequence of the biomass reaction not being able to carry a flux. Why does the biomass reaction not carry a flux? That is what we are looking at essentially. So, the other issue could be that what we just discussed, we could have dead end metabolites spoiling the show, or blocked reactions do not affect. Blocked reactions are unnecessary baggage, they exist, they do not carry a flux, but they do not affect your simulation as such. There may be mistakes in the model or gaps in the model, that is something we will have to diagnose later. But for now, why would we have 0 flux? It could be because your median constraints are wrong, or maybe there are some dead end metabolites in the model which do not have a source or sink, but typically these are two major things. The third thing could be that there is usually an additional flux known as flux for ATP, right. So, this can also go wrong. What is this? So, you have something known as growth associated maintenance, right. There is obviously some ATP that is going into your biomass, right. We did see that in the biomass equation in the previous class, but in addition to this, the cell may require some other ATP, some more ATP for sustaining other cellular function, right. So, if the cell is really decelerated like we were doing earlier, reducing the amount of glucose to a very low value and so on, maybe the cell is not producing enough sufficient ATP to sustain life. It is able to sustain metabolism theoretically, but it cannot support other important functions, right. So, this is usually bundled up as another constraint which says some V ATP maintenance greater than some thing that is for E. coli it is something of this sort. So, this is actually a fit value to make sure that your lethality results agree nicely with, or your growth rates agree nicely with experimental values.
So, blocked reactions are because of dead end metabolites, right?
Blocked reactions are because of dead end metabolites, yes.
So, second thing, you can just tell that because of block reaction. Second.
Well, not exactly. So,
That's only if it's an always blocked reaction.
so you could have, yeah. So you could have, you know, several pathways like this, and you can this could be a blocked reaction, not causing any damage. If one of these reactions is blocked, right? If this were blocked, then you have trouble because the biomass gets blocked in turn, because everything is like sort of interconnected.
Yes, sir. And in this case also the rightmost one is dead end metabolite, right?
These are dead end metabolites,
Okay,
but they are not going to affect the, so their fluxes are going to drop to 0. It is not going to affect the rest of the fluxes. But if some important metabolite here is dead end, that is where the problem comes. So all these are actually interconnected, which means that, you know, your medium was wrong or something's not been specified right. So you need to open an exchange flux for this. So you need this from outside, then it all works. And let's again revisit the concept of multiple solutions, right. We have been belaboring that over the last few classes and the previous lab session and so on, right. So this is what you are solving, right. So let us say the solution, let us say you got the V bio as V bio star. This is like the growth rate, let us say this is your 0.8739 some units that we just saw earlier in for the E. coli model, right. So now if you used a different solver or, you know, something else, what you are guaranteed to get is the same value for V bio star, but the V vector itself could be anything. You could have multiple V vectors that give the same growth rate. So this being the case, it is very interesting to understand how much variation can it admit, or rather this V is going to be something like right. So how much can, what is the leeway that V1 has for variation, what is the leeway that V2 has for variation, what is the leeway that VR has for variation. So can you think of what is the maximum value of V1 that is possible? Can you set up a simulation to figure that? What is the maximum value of V1 that is admissible by the cell?
Aren't you going to be giving an upper bound?
See, that is the upper bound, right. But what does the cell, see, we even saw earlier in one of the examples that we said that the upper bound of oxygen was minus 100 or something, but the real value of oxygen uptake rate was minus 31 or something like that.
Yeah, but that was one solution. You didn't see all the solutions.
Right. Yeah, so but that might actually be the, you do not know, that could very well have been the maximum oxygen uptake bound under those conditions. So that is what we are getting at. So how would you formulate this? How would you find the maximum value of V1? So you can basically sort of reframe your objective function. Now you do the same thing, you first do max C transpose V such that SV equal to 0. You compute your V bio star with this, right. This is the F value, optimal growth rate. Now you add a condition, V bio equals V bio star, and now maximize V1 such that SV equal to 0 and V bio equals V bio star. You can also minimize it. This is known as flux variability analysis. It tells you what are the boundaries of the flux cone essentially. We were talking about a cone, right. So you have several fluxes and there is a cone. So what is the boundary in some sense. So each flux, how much can it vary? V1 can vary between so much. So this tells you, so suppose you know that, so what is a blocked reaction in light of FBA?
It's the bound is 0. Maximum value is 0.
Max and min are both 0.
Maximum.
Right. So that is a blocked reaction, very simple way to define it.
So V bio star is 0.8739.
Correct.
You want to maximize C transpose V.
We've already maximized C transpose V to get this.
So C will be having only one value, only one in one place, and everything will be 0.
Yeah.
So whatever value of V might be, to maximize it, that particular thing is, if it is becoming more than if it is that particular element, if it becomes more than everything, then we can tell it's maximum, right? We will not care about the remaining n minus 1 element.
But they are all constrained, no? By SV equal to 0.
Oh, yeah.
So everything is intimately tied. So the almost no variable can vary freely. V bio is connected to several other things. It is connected to everything in that in this equation, right? This is your V bio, right? So it is connected to everything in this equation. So you will find that all the fluxes are actually interlinked by SV equal to 0.
So if you get V1 star, so to speak, the max V1 corresponding to that, and then now again you put it back in the model and maximize for V bio, will you get V bio star? With that constraint?
Yes, yes, yeah, that will be valid. Because we know that this is an admissible, feasible solution. So unless you say that, suppose you solved x plus y equals 10 and you got x as 5, right, and now you put that value back into x plus y equals 10 and find out the value for it, it will still be feasible, right, unless you put in an arbitrary value. Now you put minus 10 and then try to figure out or something, then you would have a problem. So this has very interesting implications, right. So now you are absolutely convinced that there are many vectors, right, which can give you the same V bio. Which of these vectors is more biologically valid? Right. So one argument was this idea called parsimonious FBA, which is you be parsimonious with the vector. So if you can drive something to 0, please drive it to 0. Okay. So which means that you minimize you can talk about minimizing the L1 norm or the L0 norm of V. So of all possible V's that solve this, can you find that which minimizes the L1 norm or L0 norm? And in fact this is built into optimized CV model as well. This is like a sparse solutions. So parsimony is the same as sparsity. So these are some very important points that one needs to keep in mind while working with flux balance models or with metabolic models and studying flux balance analysis.
But why is the most sparse vector the most biologically plausible?
Because there is always some economy in biology, right? So the cell tries to expend its resources very wisely. It wouldn't be wastefully expending reactions that driving reactions at a rate higher than it's necessary. But and mathematically speaking, you can have a few cycles or loops that are taking arbitrary values, so you can drive them to a low value or even cancel them out.
So this just like nullifies as many reactions as possible.
Yeah. And this is also somewhat related to solver artifacts and so on. So the solver will try to give you so each solver depending upon the algorithm will arrive at one of these. So this gives you a handle to get a more unique solution. It may still not be unique, there is no guarantee about that. What is guaranteed of course is still your V bio star is unique, right, but these solutions may still vary.
So if your So that problem where for bioreactor design, we had to find some optimum between the cell maximizing its growth rate and you wanting to maximize metabolite. If your answer is not too off, you can just use this. You can get maximum cell growth rate, and then find the maximum admissible metabolite and then adjust it.
Very good. So that is bi-level optimization that people normally do to kind of find the optimal rate at. So what is the max product for the max biomass? So in this video, I hope you got a good overview of how one goes about troubleshooting an FBA simulation, and we also studied the concept of ATP maintenance flux and the very interesting concept of flux variability analysis. In the next video, we will go back to how we perturb these metabolic networks and look at the other kind of perturbation. We have already looked at deletions previously. Now let us look at overexpression, which involves this interesting technique known as FSEOF, which stands for flux scanning based on enforced objective flux.